Bismillahirrahmanirrahim, and welcome back to this next video. And in this video we are going to focus on the structure of the antibody. In my previous videos I've told you about the different maturation and the developmental stages of the B cells, and once the B cells they are mature, they are going to produce the antibody. So in this video we are going to see the structure of the antibody. Now, imagine a scenario where there is a new deadly virus in the city that is affecting the whole population, and the people they are getting sick. As the virus that is new, the doctors they are very worried how to tackle this particular viral condition. Fortunately, the scientists come up with a vaccine against this particular new virus. And when the vaccine that has been given to the population, what they do is the people now they are producing antibodies. Now these antibodies they are giving strength to the people to fight this particular deadly virus, thereby neutralizing it, and the population that is getting healthy. So what are these antibodies? Let us see about the structure of this antibody. These antibodies, they're also known as the immunoglobulins. These antibodies, they are protein in nature, they are specialized proteins, and they are produced by the B cells. Now, these antibodies, they are very important in the identification and the neutralization of the foreign material. For example, it can help identify and neutralize the bacteria. They are very good in neutralizing the viruses. They are also very good in neutralizing the pathogens, and they are very important in neutralizing a condition where there are toxin that are produced by the pathogenic bacteria. So the antibodies they are very important part of your immune system. If you look at the structure of the antibody, the general structure of the antibody is usually a Y-shaped structure. If you look at this Y-shaped structure, this is made up of four polypeptide chains. And in these four polypeptide chains, you have got two heavy chains and you have got two light chains. This is the structure of the antibody. Now, let us talk about the light and the heavy chains. These two, if you look at this pink one and this green one, they are actually the light chains. And if you look at their molecular weight, this is about 25 to 30 kilodaltons, because there are different types of the antibodies. So you are going to see a variation in the molecular weight of these light chains. So this is one light chain, this is another light chain. When you talk about these light chains, within the light chains, you are going to see two domains. One, this V, this is actually a variable domain, and this C, this is actually a constant domain. So this V, when there is an L with this, that means it is representing the variable domain of the light chain. When there is an L with a C, that is actually representing the constant region or the constant domain of the light chain. When we talk about the light chain, there is one variable domain and there is one constant domain. Similarly, when you talk about these heavy chains, this is one heavy chain starting from here up to here. This is another heavy chain starting from here, moving down up to here. This is the heavy chains. And if you look at the molecular weight of the heavy chains, there is about 50 to 70 kilodaltons. Therefore, we call these chain is the light one. We call them is the heavy chains because of the differences in their molecular weight. They have got a low molecular weight, so they are light chains. They have got high molecular weight, so we call them is the heavy chains. If you talk about these heavy chains, just like the light chains, you are going to see the variable domain and you are going to see the constant domain. If you look at the heavy chains, in heavy chains, you are going to see one variable domain, and that is there is an H with the V. So this H is for the heavy chain. So this variable domain is of the heavy chain, and this variable domain that is of the light chain. So the L and the H, they are referring to the light and the heavy respectively. When you talk about the constant domain, so in contrast to the light chains, you are going to see 3 to 4 constant domains in the heavy chains, and you can see in this particular diagram they are three. So this is the constant domain of the heavy chain number one, this is the constant domain of the heavy chain number two, and this is the constant domain of the heavy chain number 3. So you can expect up to 4 and different types of the antibodies. The another important thing is these disulfide bonds and these hinge regions. If you look at the light chains and the heavy chains over here, they are connected to each other by these disulfide bonds. So this heavy chain is connected to this light chain by this disulfide bond. This heavy chain is connected to this light chain by this disulfide bond. Secondly, you have got this hinge region. This is actually the hinge region. And if you look at the hinge region, there you have got the disulfide bonds, but these disulfide bonds are responsible for the interaction between the two heavy chains. So this heavy chain is connected to this heavy chain by these disulfide bonds. Another important structural component that you see over here, that is the FAB. This is known as the fragment antigen binding. So the F is for the fragment, the A is for the antigen, and this is for the binding. So this is sometimes also known as the antigen binding fragment. And the antigen binding fragment, it starts from here and it moves up to here. What that means is that it is going to contain the variable domains of the light in the heavy chains, and a constant domains of the light chain, and one constant domain of the heavy chain. Similarly, this particular region, this one and this one, that would be another FAB fragment in this particular antibody. Now, within this FAB, the fragment antigen binding, there is an antigen binding site. And this antigen binding site is actually composed of the light chain, the variable domain of the light chain, and the variable domain of the heavy chains, whole of this one, and some part of the constant domain as well. So the whole VL plus part of the CL, the whole VH plus part of the CH1, that is actually going to give you the antigen binding site. Another important structure that you see over here is this FC. This FC is actually the crystallizable fragment, or this is sometimes known as the fragment crystallizable. Now, if the antigen binding site is present in the FAB fragment, so what is the function of the FC then? If you talk about this FC, this is very important in interaction with the cell surface receptors, which are known as the FC receptors. So there would be FC receptors on particular cells, and the antibodies, they will be using their FC regions to interact with these cell surface receptors. And these surface receptors are known as the FC receptors because they interact with the FC region of the antibody. This FC region is also very important in the activation of the complement system that we have already discussed in my previous videos. If you look at these light chains over here, so the light chains, they come in two varieties. So when you talk about the structure of the antibodies, the light chain, they are going to come in two varieties. One light chain is known as the lambda light chain. The other one is known as the kappa light chain. And in the structure of the antibody, the antibodies are going to have only the kappa or the lambda light chains, but not both. What I mean by that is that if this is an antibody, and if you are using this light chain as the lambda one, so this light chain will also be the lambda one. If you are using this as a kappa light chain, this one again would be a kappa one. So this is not going to happen that this is the lambda one and this is the kappa one. So you are going to use only a single type of the light chain in a single antibody. You cannot use a hetero form of the lambda and the kappa in making a single antibody. Now, these lambda and kappa, the ratios are also different in many different organisms. For example, if you talk about the humans, so the kappa to lambda ratio, that is 2 to 1, but in mice, this particular ratio is 20 to 1. This is the N-terminus, as we are talking about a single polypeptide. So this is going to be the N-terminus of the antibody, and this is going to be the carboxy terminus of the antibody. When we talk about the structure of the antibodies, you are going to see the complementarity-determining region as well. This is one of the important terms that usually students confuse with the antigen binding site. So you need to understand this. So the complementary determining regions, they are actually the regions that are part of the variable domain. They are not part of the constant domain. Most of this is the part of the variable domain, but some part of it come from the constant region. If you talk about these complementarity determining region, they are actually the polypeptide segments of the variable chains in antibodies. So if this is the variable chain this of the light chain, for example, if this is the variable chain of the heavy chain, for example, so you are going to see these CDRs, these complementarity determining regions in the variable region of the light and the heavy chains. Now, these CDRs, they are actually a set of the, you can say, these CDRs, they constitute a paratope or the antigen binding site. So, the paratope, the antigen binding sites, the CDRs, that actually refers to one and the same thing. And most of the time, they are used synonymously. So, whenever you see a paratope or the antigen binding site or the CDRs, you are actually talking about part of the antibody that is responsible for interacting with a particular antigen. Now, when you talk about these CDRs, there are three per variable domain. So, if this is the light chain and this is the heavy chain, what you are going to see three CDRs in the light chain, and they are known as the CDR L1, the CDR L2, and the CDR L3. So, this is the complementarity determining region of the light chain number one. This is the complementarity determining region of the light chain number 2, and this is the complementarity determining region of the light chain number 3. So in a single variable domain, you are going to see three CDRs. So if there are three in the light chain, you are going to see three in the heavy chain as well, and they are known as the CDRH1. This would be the complementarity determining region of the heavy chain number 1. This would be the CDR of the heavy chain number 2, and this would be the CDR of the heavy chain number 3. So what that means is that you are going to see a total of 12 CDRs in a single antibody molecule. You may be thinking, how come there are 12 CDRs in a single antibody? If you look at this structure, you have got one light chain over here, you have got one light chain over here, you have got one heavy chain over here, you have got one heavy chain over here. So if there are three CDRs in this particular variable region, there are three CDRs in this particular variable region, three in this particular variable region, and three in this particular variable region. So you are going to see a total of 12 CDR in a single antibody molecule, because the antibody, just I told you, they are made up of two heavy chains and they are made up of two light chains. So if there are two light chains, there are a total of six, you can say, CDR, and there are a total of 6 CDR in the heavy chain as well. So in a single antibody, you are going to see a total of 12 CDRs. Now, sometimes these CDRs, they are referred to as the hypervariable regions. So they are, you can see, the regions of the antibody, they are very variable. So you are going to see a lot of differences in these particular regions between one antibody and the other antibody. Now, this hypervariability, that is very important, because these CDRs, as they are hypervariable regions, they are very crucial to the diversity of antigen specificities. A lot of antigens, they are there in the environment, and if your antibodies are going to recognize these whole antigens, there should be variability in the antibodies, and these are actually the CDR regions of the antibody, which are very important for determining the diversity of the antigen specificities, as these are the most variable parts of the antibody. Now, how this variability that is achieved by a particular phenomenon, which is known as the somatic recombination, and we are going to have a detailed discussion on the somatic recombination, how this somatic recombination is important for the generation of the diversity of antigen specificities. So we are going to continue the discussion in the next video.